Great. Well, it's a pleasure to be here virtually to present the first public State of the Fold address. Since it's the first of these talks and a new idea, what I thought I would do is give some of the historical context about folding at home so that it's recorded somewhere, and I figure people will appreciate that, and sort of go through the past, present, and future of the project. And given how much we've done on the present, I'll focus particularly on the past and the future. So just to give some very basic background to make sure everyone is up to speed, we at Folding at Home are really obsessed with proteins and protein dynamics generally. And what a protein is a chain of chemicals called amino acids. So I'm showing an example here. They have a common set of atoms that we call the backbone, and then each is decorated with one of about 20 different choices for this R group here called the side chain, right? So you have this linear chain of amino acids, and somehow the way that these chemicals push and pull on each other allows many proteins to spontaneously fold up into a compact three-dimensional shape that's capable of performing some function. So oftentimes we like to show these structures as a ribbon diagram like this here. So this is very useful in that you can see, where the two ends of this chain are, and this ribbon just tracks the backbone. So you can get an overall sense of what this protein looks like. It can be a little bit misleading though, right? Because to the non-expert, you can come away thinking, wow, there's a lot of empty space in proteins. They're these pretty, funky looking objects. So if we instead switch to a view where we have a little sphere for every atom in the protein, you can see that this is still a funky looking little object. It's on a much smaller length scale than we're typically thinking about on our macroscopic length scales. But this is a pretty compact globular shape, right? And so there isn't all this void space, and it's tempting to think of these things as rigid bodies whose shapes allow them to perform some function. Right, so in the case of this protein, you can see this sort of pocket here. That's what we call the active site of this enzyme. And that's where this protein is able to bind to specific small molecules and perform a chemical reaction on them. And that's all, attributed to the structure that has been solved via experimental techniques like X-ray crystallography. And yet we know from the laws of physics that these things are not rigid bodies, right? There's a lot of motions going on, and one of the biggest and most dramatic motions that has attracted the curiosity of many, many scientists across the decades is the way that these things fold up spontaneously in many cases, right? So the protein folding problem that initially motivated the creation of Folding at Home, ergo the name, is that you have this linear chain of chemicals called amino acids. They're produced as a linear chain like this. And like I said, somehow the way that they push and pull on each other allows them to fold up into these reasonably specific, very compact shapes that can perform some function. And this is just mind-blowing that they can do this all on their own, oftentimes without any input from any external source, right? It's kind of like if I said, hey, I'm going to dump a bunch of car parts on your front lawn, and they're going to assemble themselves without you or an engineer or anyone interfering. They're going to assemble themselves into a working Porsche. And you'd say, wow, that's awesome. And maybe sometime I'd like a Ferrari, actually. So I'd really like to understand how that works and be able to manipulate the process. And one little nuance here is that people will talk about the protein folding problem, but there's really a variety of problems, right? And two of the biggest are, can you predict, given the sequence of amino acids, what this dominant structure that is typically seen experimentally is going to look like? And the other part of this is, how do you get from this extended chain to this compact shape, right? It's not teleporting, there's some physical process. And Folding at Home has always been interested in particular in this physical process of folding. And part of the motivation has always been that if we could understand how proteins undergo this extremely dramatic conformational change or change in their geometric shape, then we're set up to understand all kinds of interesting questions like how the moving parts of a folded protein allow them to act as toggle switches, or how the moving parts allow them to perform catalysis and make chemical reactions occur many orders of magnitude faster than they would without the participation of a protein. So a very big challenge, and experimentally, there's no way still after decades to watch this folding process with the sort of atomistic detail that one would want to see how changing one amino acid, so just tens of atoms out of many thousands, how it changes this whole process, because we know it does in many cases, right? So what we really want is a way to watch moving processes like this. And one of the very powerful opportunities is to use computer simulations, where we say, okay, it's going to be a long time before we're ever able to, if we're ever able to develop a technology that is the experimental equivalent of a microscope, where we can zoom in on a protein and then watch how all the atoms move. But with the power of computers, we can simulate this process. And so the idea is to put our protein, again, with all the atoms represented as spheres here, in a box of water and salts and whatever else, is reasonable mimic of the protein's natural environment. And then we're going to keep asking over and over and over again, how is every atom in this protein and its surroundings pushing and pulling on every other atom? And by doing this, we can watch how these things move over time, right? And so this is just a little teeny tiny snippet of simulation. Don't blink, you'll miss it. But there it is, right? And the brevity of this little snippet of simulation that I'm showing a couple times here, but haven't looped just to highlight just how short it is that these calculations, while potentially powerful, are extremely computationally expensive. Right. So in order to watch atoms, approach each other and collide and bounce off, when we ask where is every atom going to be some small time in the future, based on how it's being pushed and pulled on other atoms, that small time that we're projecting forward in time is on the order of a femtosecond, so 10 to the negative 15th seconds. Right. And where we want to be is watching processes that are biologically relevant and on like seconds and slower timescales often. So that's a lot of orders of magnitude in time to build up. And this is an old slide. I stole it from my PhD defense in 2010. But down here, these aren't terribly old numbers. They're a little outdated, but it can take a long time to do that many iterations of simulation, right? For lots of the things that we want to see, if I was to tell one of the students in my lab to do it, by brute force on their, nice desktop that I've purchased for them, in order to get the data that they need to even start their addressing their research project, they would first need to build a time traveling device and go back to, when the pyramids were built. And put that machine in a safe place with a really great power supply, because they've got some waiting to do, right? So obviously this is not a tractable way to make scientific progress. And so folding at home was born when our founder, Vijay Pandey, in 1999 was starting up his new lab at Stanford University and thinking about, well, how are we going to tackle these really hard problems that are computationally tractable? And so looking for inspiration, he and many had been fascinated by phenomena of the time that many may remember, and many of you may have not heard of, which is a software package called Napster, which was one of the first big platforms for peer-to-peer file sharing, right? And so people were using this for sharing music and all kinds of other things, of course, but music was the poster child in my recollection. And Vijay just thought this was fascinating and had always been fascinated with computers, and had done his training and work in theory and getting into the potential for computers to allow us to do things with theory that no pen and paper will ever achieve. Because of just the sheer number of calculations required to numerically solve some of these problems. And so taking inspiration from Napster got him thinking, wow, people with computers can do a lot of cool things working semi-independently, but also kind of together. And could we take inspiration from that and come up with a computing paradigm that allows us to make progress on these really intractable problems, right? Because at the time, much of the focus of the simulation community, which was pretty young at that point in the grand scheme of things, was on, okay, well, how, if one computer can't simulate these things in a reasonable amount of wall clock time, then how do we get lots of computers in the context of a supercomputer working together on this? And that's obviously, got huge value and is very, very useful, but, you can only get so far, right? And the issue is that, no matter how much money you have, Bill Gates can get a bigger computer than I can and somewhat faster computer, but he can't get, 10 orders of magnitude faster than my computer because money just can't buy that. We don't have the technology. And so what can we do? And so one of the common things to do at the time was to think about protein folding as a sort of dart throwing exercise, right? You throw a dart, which is a trajectory. It's going to travel through space and time, and it's either going to hit the bullseye, it's going to end up, in the folded state of that protein, or it's not, right? In contrast to my dart analogy, this is really a stochastic process, right? And so the idea with running one big long simulation is, okay, we're gonna, have this dart and we're gonna throw it, and having been thrown, it's gonna fly around for infinite time, zigging and zagging. And what we want to do is run this simulation long enough that it reaches that target, right? And it will eventually, the laws of physics tell us it will, and what we know experimentally, as long as the simulation is a reasonably faithful recapitulation of reality. And so the problem is, how do we get enough computers working together to make the simulation go fast enough that we can get to that target in a short enough time that it's reasonable for someone to do in their PhD thesis, for example. Right, and like I said, a huge amount of valuable work has been done on this. This is not something I'm trying to trash by any means, but there are other strategies, right? And so the strategy that Vijay and his early graduate students devised was to say, well, look, really, if we had a long enough trajectory, this is making multiple independent attempts at getting to this target. And so given that, we could run this one big long simulation that takes an attempt once, then takes an attempt twice, takes an attempt 3 times, on and on and on until we get a success. And then we can see, what it did, or keep running and try to build up multiple successes, right, to get some statistics on the stochastic process. But if these are really independent anyways, why not do them independently? And so what they did was sat down with their pens and papers, and they devised a scheme where you could, instead of running one long simulation, you would start many shorter simulations that would be run in parallel. And instead of saying from my one long simulation, I tried to fold 1,000 times and I succeeded 10 times, and learning something about the protein folding process and the rate of reaching that folded state from that serially gathered data, was, well, let's run 10,000 attempts in parallel, right? They're each going to be long enough to be a single attempt. And then we can gather those same statistics, and we can say, oh, I tried 1,000 times and I got 10 successes out of those. And now I can focus in on those trajectories and see what they did. And, based on how many successes compared to the number of failures to reach the folded state, I can infer the protein folding rate. And this will be a great approach to science. So with these theoretical tools in hand, then what they did is they went and started folding at home, right? So this was the relatively early days of the internet. And so you can see their initial computing setup didn't look quite like what we're accustomed to today. And it was a really heroic effort to put this together, and it took a lot of babysitting to keep it going. There's lots of stories about coming in at odd hours to deal with funny computer problems and keep this alive. But in the end, what this allowed Vijay to do was to set up this system called Folding at Home, where you can queue up simulations on the servers that are run by the research group. and ask people, with computers and an internet connection, anyone who wants to install our software on their personal computer or computers. And what this does over and over again is reach out to our servers, say what hardware the volunteer has available and that it's available to do work. And the servers will match, the volunteer's hardware with an appropriate chunk of simulation from the queue. Send the volunteer the initial structure of the protein to start from and all of the relevant parameters and how long to run. And then the volunteer goes off in the background and runs that chunk of simulation, asking over and over and over again, where is each atom in this protein going to be some small time in the future, given how they all push and pull on each other. And at regular intervals, the coordinates of all the atoms are saved to build up a little snippet of a movie, basically, that we call a trajectory or a simulation. And that's sent back to the researcher servers, and then the volunteer's computer will volunteer for another chunk of simulation that we call a work unit, right? And once a work unit comes back to the servers, the last snapshot on that work unit will be taken and used to generate the starting point for a new chunk of simulation that is then added to the queue, right? And so at the end, you get all kinds of simulations, like 1,000, 10,000 that are run in parallel. Each is broken down into a bunch of work units that are consecutive snippets of a longer movie. And on our side, we can start taking that data and making inferences about protein folding. So the initial way that this was done was with this very simple view of the protein folding world, which is that the protein is either folded or not folded at any time. And given these trajectories where you can say, is it folded? Is it folded? Is it folded? Is it folded? You can now, ask, when does it get to the folded state? And then you can look back and ask, well, how did it get there? How often does it get there? All kinds of interesting information. But this very binary paradigm is still limited. And so the group kept thinking, well, look, there's a lot of conformations out there. We know it's not so simple as folded or not folded. Even within the folded state, there's all kinds of interesting dynamics going on. And so what the lab started working on and was just getting into really when I joined the lab in 2006 was, how can we take all this data and essentially build what a map of the conformational space that we explore with our simulations. So now there aren't just two states, folded and unfolded. We can have an arbitrary number of states, as many as we need. You can think of these states as like cities on a map, right? And what we want to do is use our simulations to identify the structures that a protein is prone to adopt and the rates of hopping between adjacent sets of structures, right? And so this really is like a map, right? You want to go build a map of the United States. You need to go find out where the cities are. You need to figure out cities and towns, which are connected by roads and which aren't, and how fast can you go on those roads, right? And with that information, you can now, build a new maps app, for example, right? And so there's many ways that you could go about, getting the data that you need for your maps app. Sticking with the map analogy, you could say, well, if there was no, central registration of everything, I just need to go explore, right? So I could spend a lot of money building the fastest car that I could, and then I can go zip around the roadways on this car, enumerating what's out there and how quickly you can go on roads. You could figure out how quickly you could go with a normal car, and so you could serially go about exploring this space and building your map, right? But we all know that that would be horribly inefficient, right? Again, Bill Gates can buy a much faster car than I can, but he can only go so much faster. He can't get one that's a billion times faster than what I can buy, since he has a billion times more money, right? It's just not physically possible given what we know about physics and the state of technology, right? And so the far more efficient way to build a map is to say, okay, well, I'm gonna get a bunch of people to go out to drive around in their cars, and I'm gonna have them send me back their GPS coordinates as they commute to work and go to the grocery store, and so on and so forth. Some of them are gonna go on road trips or, go visit grandma in a different state or whatever, right? And now I'm going to take all this data, and as long as there's overlap between the space that is explored by people, you can piece together this global map that captures events on length and time scales that are far beyond what any individual explorer is going to do, right? And so that's the approach that we take with this, conformational mapping exercise using methods that we call Markov state models. Let's send out our explorers, thousands of simulations, ideally started from different initial points, and we're gonna, get back these GPS coordinates, these structures of the protein stored at regular intervals during these simulations, and we're gonna infer this map from all this data. And this has been extremely powerful, but it was tough going, right? When I joined the lab, John Cadera and some collaborators had just published a really nice paper where they made this work and put out a software tool for building these Markov models from simulation data. And they worked on a number of different systems. So, John, please don't get mad at me for focusing on the simplest one that you attacked. But the one that was the focus of the paper was this system called the alanine dipeptide, right? And this is basically a single amino acid from a protein. And it's got these two bonds that can rotate that you can see here. So there's only two things that can change, right, from a single amino acid, whereas most proteins are made out of hundreds of amino acids. And some of them, have many thousands of amino acids, right? So this is definitely not your typical protein system. So this was a very, very important step in the evolution of these tools, but there was a long way to go, both in terms of the computational power and our understanding of how to build these maps and make the best use of the data and so on and so forth. So without going into the details, John continued working on this. I got involved. Vince Bowles got involved. Shuhui Huang was another major player. There's a bunch of others, but those are some of the people that are, I want to highlight because they're still involved with folding at home. And we made some really significant progress. So, beginning of my PhD, paper focusing on the alanine dipeptide, which has motions on like a picosecond time scale, so very fast. Towards the end of my PhD, Vince and I published this paper where we were able to fold a protein with 35 residues that on average took a millisecond to fold, right? And this was a big deal because prior to that, it was a big deal to fold a little 30 residue protein that's barely safe to call a protein that folds on a microsecond time scale. And that was hard, right? So this is 1,000 times harder problem than the rest of the field is effectively even trying to touch that we're bringing folding at home and these Markov state model methods to bear. So I'm going to take a couple minutes and let this video speak for itself. But just as background, I thought it would be funny to share that the impetus for this video was that Vijay went and gave a talk on what we had done. And being a quantitative scientist, he had really focused on the statistical aspects of what we had done, how it had worked and how good the error bars were and how this was really the way to do things. And one of his colleagues sat him down afterwards and said, Vijay, I'm with you. I love it. I think that, in my ideal world, science would be that way. But, even in science, most people don't work that way, right? And what everyone wants to see, even me is like, Vijay, I want to see the God view, right? I want to see how this protein folds as if I was God and I could zoom in and I could watch it happen. So Vijay came back and bounced around some ideas with Vince and I and others, and he put together this movie. So again, I'll let it speak for itself for a minute. All right, so there it is, the God view of NTL9 folding into its stable folded structure and then staying there afterwards as it should for a prolonged period of time. I don't know if Zoom took the music through and if you could hear that, so we should splice it in after the fact if it didn't, because it adds some nice drama to the event. But I love watching this thing. It was so cool. And there's another funny story for those of you who are in graduate school or who are postdocs. We had this big initiative that again, John and Vince and Shuhui and I were all involved in, where we were going to try to fold a bunch of different proteins, including NTL9. So we set up all these simulations and had them going on folding at home. And I remember following up with Vince and being like, "Hey, I wonder if they folded. We'd left them for a long enough time that it was possible." And I remember, I think it was not a great day for Vince because he was kind of like, "Yeah, that's not going to happen, Greg." Like, was effectively the message. But being one of the newer members in lab and having some bold naivete, I went and looked at the data, and we had folded NTL9. It had gotten within a couple angstroms of the crystallographic structure. And so I think, this is, one of many early lessons for me that if you have enough simulation power, these simulations really are extremely powerful and can provide a lot of insight. All right, so since then we've been building off of this and going in all kinds of directions, right? The very early focus of the Folding at Home project was, all within Vijay's lab, since that's where Folding at Home was born. He had the foresight to tell people that if they went on to academic careers, that they were welcome to stay involved with Folding at Home as long as they were giving back to the project somehow. And so that was really wise of him, keeping people like me and the others I keep listing involved. So the initial focus was on the technology to make this work and insights into protein folding. And as we've really made tremendous headway in that area, a number of us have been switching our attention to questions like, well, how is it that these proteins actually work, right? How do they work as toggle switches? How is it that mutations alter the behavior of a protein to give rise to cancer or Alzheimer's disease? And how can we, use this insight into proteins' moving parts to inform the development of new therapeutics? So just to give a sense, even within my own lab, we're working on all kinds of things, right? Prior to the COVID-19 pandemic, we had a major investment in looking at viral proteins, like this protein called VP35 from the Ebola virus. And the idea was to say that if you look at the static structure, the single snapshot of this protein, this essential component of the virus doesn't look like a good drug target, right? There just isn't a good spot for a drug to bind where it would interfere with the function of this protein. And so people really haven't gone after this protein specifically with drug design tools. But coming from this perspective of ours that protein dynamics is everywhere, we wanted to ask, well, like what sort of drug design opportunities does considering the moving parts of this protein open up? And I won't go into the details, but it does provide a really cool way to go after what we call a cryptic pocket in this protein, where the structure opens up spontaneously to form a potential binding site for a drug. And in addition to seeing that in simulations, a third of my lab is experimental, and we actually have experimental data that corroborates this prediction that we made. And we're working to target this pocket with something like a small molecule drug to show that this is really a viable drug target. All right, so we were also working on this protein called ApoE. Turns out that ApoE is the single strongest genetic risk factor for Alzheimer's disease. So if you have a single amino acid in this protein changed out of 299 amino acids, your risk of developing Alzheimer's disease can go up by as much as 15-fold. And we have no idea why and no idea how to use that insight into the potential importance of this protein in aging and dementia to inform drug design. And in fact, we haven't even been able to see the whole protein. We can only see half the protein with these existing experimental techniques like X-ray crystallography. And the hypothesis is that the rest of the protein is so floppy that we can't get it to hold still long enough to take a photo effectively. Right. And so this is a big opportunity for simulations because we don't need things to be rocks in order to study them. We can hypothetically deal with these very floppy or disordered systems. And a third is a class of proteins called myosin motors. And these play a broad range of functions in biology. You'll be most familiar with muscle contraction. Because there's so much myosin in muscle, these were the very first proteins to be studied. And we still don't really understand how they work. It's really fascinating. We know that where it binds to another protein called actin over here somehow is communicating with where it binds to small molecules like ATP and ADP over here, and that catalyzing a chemical reaction here, controls the affinity for actin over here and controls motion of this lever arm over here. And much has been learned, but we still don't understand the system well enough to say, predict a priori how changing some of the amino acids in one of these proteins is going to change how fast they move, or if they move, or how they're going to respond to being under force. And so these are really fascinating proteins, and they're also very medically important. They're some of the most mutated proteins in human disease. And so there's huge interest in being able to say, okay, well, we found a new variant in a patient with such and such disease, and it happens to fall in a myosin motor protein. So is that mutation breaking things, speeding them up, doing nothing? People would love to be able to predict that, and we just don't have the data or the tools to do it right now. And people would love to be able to design drugs that target some myosins and not others, so that you can treat heart failure without having adverse effects on other systems, for example. So I'm not going to go into great detail on all of these, and there's many other projects on things like cancer and peptoid design and how RNA is transcribed, all kinds of interesting things that we're doing on Folding at Home. But I just wanted to convey that this is really a very general platform, and we're now taking it in many directions in parallel and having impact on many different fields. But this got a little bit disrupted, coming from the past to the present by the arrival of our good friend and colleague, the SARS-CoV-2 virus and the corresponding COVID-19 disease. Not surprisingly, given that we were already working on a number of viruses, and we're just about to put our first drug that we discovered could be repurposed to treat a different viral infection. We were just about to put that into clinical trials, which was a hugely exciting step to be a part of, to see this project going from this very basic focus to someone actually ingesting a drug, in part because what we had learned from computer simulations. It was amazing to have been a part of, right? But this all got put on hold by the arrival of the pandemic. And it didn't take us too long to realize that, hey, we should take our general platform and pivot to apply all of these tools to see what we can learn about how the virus functions and how we could inform the development of new vaccines and therapeutics. And so I want to give a particular shout out to Rafael and to John for being the first to see the opportunity and launch some simulations on Folding at Home. The rest has been a truly astounding ride, right? At the beginning of 2020, before the pandemic, we had about 30,000 active devices participating in Folding at Home. Within the first couple of months of launching our first COVID-19 simulations, we'd increased that number to about over 1 million active devices running Folding at Home at a given time. And the upshot of this is that Folding at Home once again became the world's fastest computer. So we were the first to reach the exascale. So exaflops being unit of measure. So folding at home was also the first to reach petaflops, and so it's cool that we were the first to reach exaflops, even with a very conservative estimate of the raw compute power that folding at home had to offer with the help of all of our volunteers. And it exceeded the world's fastest supercomputer at the time by at least 5-fold, right? So truly amazing resource. And I'm not going to spend a lot of time on this. I've given a bunch of talks on this, as have others in the folding at home community, and many are on YouTube. But just to give a sense, one of the things that we were very interested in my lab was this complex of 3 proteins called the spikes. So each of these red protrusions in this canonical image of the virus is a spike complex. And these things have to open up in order to expose the part of the protein that grabs to a human protein called ACE2 and to initiate infection of a prospective host, right? So this motion has to occur, but mostly what has been seen experimentally is the closed up structure of this complex. And so we really wanted to see what it looked like for this thing to open up and what the open state looks like and what information that could provide. And this is a big deal, right? The main component of this complex has 3,600 residues, right? So that is 3,600 times the size of the alanine dipeptide that we were publishing on in the mid 2000s, right? So it's quite a big step. And so here's one of these spikes. There's 3 proteins that are identical to each other. They're each colored a different color. There's this ribbon diagram for each one. And then in transparent surface is just showing the space-filling representation of this spike. The upper left is where this thing is tethered to the membrane. The lower right is where it has to open up in order to have the potential to bind to this other protein called ACE2. And because of this symmetry at this end, when we were envisioning this before having any data on what opening looked like, we were envisioning it like the opening of the mouth of one of these demogorgon monsters from the television Stranger Things. So we've taken to calling this thing the COVID-19 demogorgon. But if you just run, even big simulation by the standards of today, 10 microseconds long, all you see is some wiggling and jiggling around the starting state, but with the power of some simulation technologies that I'll touch on shortly and folding at home, what we're able to see is some really massive conformational changes. So here you can see one of the chunks of this protein largely dramatically dissociating from the rest of the protein complex, creating this huge opening and exposing all kinds of buried surfaces, including the part of this protein that, as I keep saying, is responsible for binding to this human protein called ACE2 and initiating infection. Right, so this has all kinds of interesting insight into, why the SARS-CoV-2 virus has been such a problem for us when other coronaviruses aren't. Why antibodies are able to bind the spikes of some coronaviruses but not others. And I hope that one day it'll help inform, the future efforts to keep our vaccines ahead of the virus in terms of the inevitable arm race that will exist between us and the virus. So again, there's a lot that can be said here, but instead of going deep on that, since we've done that a number of times, what I want to mention is what's coming next, right? And so one of the things that we're trying to streamline is that we're generating some insane amounts of data, right? In a typical paper in our field, people will have maybe a microsecond of simulation data for one protein, right? And in our first preprint, which is now finally accepted for publication and will be coming out, having been peer reviewed soon, we have over 60 different proteins and complexes that we looked at. Instead of having 1 microsecond of data, we have over 0.1 seconds in aggregate of simulation data. And it is a huge data set, over 200 terabytes. Right, so this is not something that we can just, put on our little wimpy web server that hosts our website and let anyone download. And so we put our Markov models on a platform called OSF for anyone who wants to download them. And we've been fortunate to receive support from AWS and their public data sets program. And so they are hosting all 200 terabytes of the raw simulation data for us on their S3 platform, so that anyone who wants to can download the Markov models is what I would suggest, and you can play with those. They're a good platform for all kinds of analyses. But if you're a hardcore methods developer, or want to look at the data a different way, you can also get the raw data. So this is one of the things that I think is a really nice step for driving scientific progress, is making it as easy as possible for as many minds as possible to crunch on this data, because there's so many different questions you can ask with it. So another thing that we're doing a lot of is developing new techniques that draw on what we've learned from all of these massive simulations that we've run. And again, I'm definitely overrepresenting my lab's work, and that's not to undervalue anyone else's work. It's just what I know best. So just to give one example, we have improved ways to do what we call adaptive sampling. And so the idea is to say, okay, well, ultimately we want to build maps of these spaces, right? And right now, the way we do that on Folding at Home is we launch 1,000 simulations from some initial structure, right? So you imagine we want to build a map of this landscape where blue is valleys and red is mountains, and mostly what we want to see is the valleys, right? We have some starting points, so we know one point on this landscape, like our kind of analogous to our crystal structure. And we know that we want to solve a particular problem, right? So my lab is very interested in going and hunting for these cryptic pockets that aren't present in the crystal structures, so our starting points, but that are present in structures that the protein fluctuates to, that have larger pocket volumes, right? So we can imagine that everything over here has larger pocket volumes than our starting structure, and we really want to see what those look like and see if they might be useful for drug design, for example. But there's also all kinds of other structures over here that don't have pocket volumes, right? And those might be important to see for solving all kinds of other problems. And in the limit of infinite compute power, I would love to have a map of the entirety of this landscape. But given that even with the power of Folding at Home, we're still compute limited, I would much rather spend all those compute resources exploring over here, right? And so what we do in our adaptive sampling algorithm called FAST is we start a set of simulations from our initial structure. We're going to let them run for a limited amount of time so they can explore locally. Now we're going to build one of these maps, one of these Markov state models from that data. And then we're going to use that map to decide, well, where on the map that I've built so far should I start the next batch of simulations in order to expand the map and improve the quality of the map with the most efficient use of my finite computing resources that I can, right? And so what we're going to do is take all of the states, all of the nodes in this network of this map that we've built, and we're going to rank them. And that ranking is going to have two components, right? It's going to have a geometric component. So that's going to say, okay, I want things over to the right. So I'm giving things, in this case, that are further to the right and have more open pockets, higher scores than things over here, right? And so that's going to promote moving to the right if the simulations can, right? And so the individual simulations don't have any bias. They're just going to wander around randomly. It's like getting on the road and flipping a coin to decide whether to turn. I guess you need a dice because you need to decide which way at each intersection you're going to go, right? And instead of having any specific objective, but we're going to control where data is collected by controlling where the simulations are started, right? So we're going to prefer starting simulations from states that we've discovered that are further to the right in this case. And then the second term is a statistical component, right? And so what that's going to do is try to prevent redundantly simulating the same states over and over again, right? Because you can imagine that if you're on this landscape and you're happily going rightwards and you get stuck here and you're surrounded by mountains that are hard to climb over, what you want to do is go backwards and try to go around that, right? Not just sit there and keep trying, which might literally take 1,000,000 years of simulation to wait until you randomly climb up this mountain and get across it, right? And so the other component is the statistical component that assigns higher ranks to states that I don't have much data for than states that I have lots of data for, right? Because if I have lots of data, I can pretty well guess what's likely to happen if I start more simulations there. Where if I don't have much data, who knows? Even if the structure is further left than other structures, starting a simulation there might surprise me and find a productive way to make rightwards progress. All right, so here's just an example then where we're starting a batch of 10 simulations from this initial structure. Those will be in a yellowish color, and then we'll color where they visited in dark green, right? And so you can see these things are exploring, they wander around randomly near the starting point, but you can see this rightwards progress. Then they get stuck at this barrier, like I said, and manage to backtrack and circumnavigate it and make really substantial rightwards progress, right? And just to contrast that, if we on the bottom just start 10 simulations and let them go, no adaptive sampling, you can see at the beginning, it's hard to tell the difference. They both go rightwards, they both get stuck, they both happen to back up. But these simulations have no intrinsic desire to go to the right? So you can see at the bottom that our non-adaptive sampling simulations end up wandering off to the left, and they bounce around and redundantly do the same stuff over and over again. And they're giving us information on part of the landscape that we already know in advance we're less interested in. Right, so we're getting orders of magnitude speed up from these techniques in terms of getting the data that we actually want before we get the data that we're less interested in. And now we're hard at work deploying these methods on folding at home. So that will be coming soon. So far, we've been running these adaptive sampling simulations on our local cluster, and then following up with simulations on folding at home to improve upon things. But more computers is better is an important message of the folding at home initiative. So getting this on folding at home would be great. So technology like this is definitely a major focus. Another big one that's really been driven by John Cadera and Vince Bowles is getting free energy calculations or chemical free energy calculations up on folding at home. And so the idea is that if you have a chemical compound that you think might bind to a protein and act as an inhibitor, one of the important things you want to know is, well, how tightly does that compound bind, right? And this is something that people have worked for a long time to develop physics-based methods to quantitatively predict. Like our simulations, hunting for cryptic pockets, these are computationally expensive methods based on the same sorts of simulation technologies. The upshot is that when people do this sort of thing, and publish on it, they're often running pretty short simulations for something on the order of a dozen compounds, and looking at how well these methods perform. But John and Vince now have these methods deployed on folding at home and have been screening tens and hundreds of thousands of compounds in parallel, right? And so this is an exciting tool to have in our tool belt, because it opens up the possibility of doing big screening campaigns against targets of interest, and either to have some experimental efforts of our own going in parallel to test with the compounds that we think should bind the tightest and see if they work experimentally, and or engaging with collaborators, which is always a really great way to have lots of good science going in parallel that leverages the expertise of the rest of the scientific community. So that's another big thing that we're doing. An important part of this, here's a little free energy simulation. An important part of this is help from our friends, right? So one of the silver linings of this horrible pandemic that we've had to endure is that folding at home really attracted a lot of attention, and we really got to show off what we can do in terms of the progress that we made in understanding how this virus works, and through the COVID moonshot collaboration, that we can actually inform drug design efforts in a meaningful way. And so it's super exciting, and John has given a number of talks on this that I think are online. That's really been, collaboration has really been his baby, but it's super exciting that there are compounds that work in biochemical experiments, and now they have data showing that they work in animal models, and are talking about what other tests needs to be done before we can start having human beings take a drug that folding at home played a role in helping to develop to tackle literally one of the biggest global health threats that we face. This is amazing progress, and I'm so proud of everyone in the folding at home community for helping out, and so grateful to everyone, and including the many organizations that have reached out to help us leverage new technologies and to leverage their in-house expertise in order to scale this effort as quickly as possible. And this has really opened our eyes to an opportunity that we hadn't been capitalizing on before, which is that there's a lot of opportunities for synergy with these companies. A lot of them, especially in the pharma world, are increasingly interested in the power of computation for informing drug discovery. And so I'm not going to go into specifics right now, but we have a number of collaborations that we'll be making announcements about. Some that we've already announced, where we've been working with some of our new collaborators to improve things like the stats website, for example, that many have seen recently. But we also have some collaborations with more pharmaceutically oriented companies that we're working on. And I want to be very clear that no one is going to be making money off of folding at home. Right? We have no interest in selling compute resources to anyone. That would be a gross abuse of our volunteers' generosity. Everything that we are going to do is going to stay in the open. All collaborations will be things where we're working together with pharma companies on developing new tools. So how do we make adaptive sampling as the greatest tool that it can be, for example? And towards that end, we're going to apply it to some of the systems that are well studied or of great interest in the academic community, and all of the data is going to be available to anyone who wants it as it's generated, is the plan, right? So there will be no question that anyone's doing anything sneaky, and I hope that we'll keep your trust and that you'll help us to do this, right? Because success for folding at home is partly driving scientific advances, but I think it's fair to say that even those of us who are really, really passionate about basic research also want to see that bear practical fruit. And we as a community are failing if we don't take the opportunity to actually show that this can improve the drug discovery process, and to share those tools and encourage the people who are actively engaged in drug discovery efforts to use those tools in their drug discovery campaigns. And we'll leave that to them. That will not be on folding at home, but we all want that to happen, right? Where I am often asked, what has folding at home done? And I would think it would be a dream come true if I could say, pharmaceutical company X, used what we learned from folding at home, and people are now taking this drug as a result. So that would be great. So we're also expanding our developer team. I want to give a huge shout out to Joseph Coughlin, who started working on folding at home while I was in graduate school. So prior to my involvement in the project, folding at home was the infrastructure for running these simulations was written by Vijay and his graduate students and postdocs, right? And once they proved that it could work, one of the interesting ironies is that folding at home, the infrastructure became less of an interesting science thing, right? It's now a software engineering problem. It's not the best thing for graduate students and postdocs to spend all of their time on, because that's not what's going to advance their career. And most of them don't have professional background in software engineering, right? So Vijay brought in Joseph, who does have a wonderful background in software engineering, who has been really driving the development. And is now the lead developer, and has been for quite a while, a lead developer of folding at home. But for most of our history, lead developer has been kind of an ironic title, because he has been an army of one, right? Joseph has done all of this on his own. We have a great team of scientists who are using folding at home and make some contributions to the infrastructure, but the vast majority of it is Joseph's baby. And the fact that we were able to scale so quickly as we went from 30,000 volunteers to over 1 million devices in a very short time scale is a testament to the quality of Joseph's work. But no matter how awesome Joseph is, he's still one person, and it would be better to have more talented people contributing. So we're working to get the resources to expand our developer team, and have been very happy recently to welcome Emma Matthews to the team, who also brings a great software engineering background and is helping us to do a bunch of things like engaging with our community more, because one of the issues with having a small team is we have very limited bandwidth for tackling all of the potential issues that we could be tackling and all of the cool additions that we could be making to folding at home. And so we have a lot of opportunities here. And so we're doing a lot to capitalize on this. I'm not going to give an exhaustive list, but we're really making big efforts to invest more in our community. So Joseph has turned a lot of his attention to a new version of our client code that will have a number of technical improvements. But one of the big things that we're doing is we're taking out all of the security concerns that prevent us from sharing the code with the current client software that's on our volunteers' machines with anyone who wants it, right? And so the plan is that this will be completely in the open source, and that other people will be able to contribute or even to make their own client software that uses our APIs to communicate with our servers. And so this is one of the things that ties into a larger effort that Emma has really, and Anton Thainel have been spearheading, which is to really engage the open source community more. We're a little bit limited in what we can open source right now because of these security concerns, but Emma and Anton have started off, and with Joseph's help, a new GitHub repository where we're already getting people involved in a number of open source initiatives. And things like this client, I think we have a great opportunity to expand that, and with more people and their passions, will I hope we'll have enough bandwidth to really make that a pleasant experience. Let me see what else we have here. Emma and others all also work together to launch a Discord server that provides new opportunities for our community to talk with each other, and the volunteers to talk with the scientists. And so this is a really great way for people to engage. We also are getting help from some of our friends. So in VMware, for example, has helped us to update our stats website, which is really great, right? We haven't had the bandwidth to do lots of things like that, and it's wonderful to have help from friends to make these sort of improvements to our infrastructure that are not essential from the perspective of the science, but really good for making folding at home a stronger community. And then a final thing that I'll mention is these talks. So I want to say a special thanks to Raffle again, and to Sukrit and to Matt Hurley for really taking the initiative to help come up with this. But we're also investing in the scientific community of folding at home. There's over a half a dozen labs from across the globe who are involved now. We've largely been operating independently, but the working together on the pandemic has really demonstrated the power of bringing more passionate and hardworking scientists together. And so to build on that, what we're doing is having monthly talks for the science side of the folding at home community. And the idea is that 2/3 of those talks are just going to be for the scientists. We're going to nerd out on the tools we're developing and share updates on what we're doing in a setting where we don't have to worry about revealing a security issue or something that should be kept private, and can really put our heads together on shared infrastructure and problems that we're all facing, for example. But we really do want to engage with the rest of our community. So the plan is that once a quarter, we're going to have someone give a talk that is intended for the broader folding at home community, trying to explain, one of the projects that we're using the folding at home infrastructure for, And so this is the inaugural of these open science talks, and I hope you've enjoyed it. We'll be posting it to YouTube and other platforms for anyone who wants to watch that missed this or is interested in rewatching parts of that. And so with that, I'd like to thank absolutely everyone who has helped with this. The last year, obviously, I would never wish for a pandemic. And if I could snap my fingers and have that extracted from our shared history, I would do it. But like I said, one silver lining is that it's been amazing, working with this group of scientists who have really dropped everything at some personal expense in order to help tackle the pandemic. And it was truly heartwarming. I don't know how to describe it, watching how many people from around the world piled on to help with this effort. And, the sheer number of people from every walk of life who got involved. And it felt like a big family reunion. There were all kinds of people who had been involved in the project in the past. There's obviously all kinds of people that had never been involved in the project before, and it was just awesome to know that we helped some people. At a minimum, we helped some people cope with feeling powerless in the face of this pandemic by giving them something to do to take action, and that this is really bearing fruit. We learned a lot about the virus. There's new drug candidates that are actually going into testing and could really make a literal, not earth-shattering, but earth-saving impact on the globe. So we'll see if we get there. This is science. We can't guarantee, but how close we've come, I think, is a strong testament to the power of this platform and the opportunities that we have looking into the future as we continue to build on this momentum. We got a lot of funding from different sources that was instrumental to the project surviving for 20 years, not just surviving, but thriving. And as I said, now we have a lot of new friends that bring a lot of interesting expertise and resources to the table in many forms. And looking ahead, I'm really excited to engage with them, and I think we have a great opportunity to accelerate the science and do compound that acceleration by improving the fully at home infrastructure, both from the scientist perspective, but also from our volunteers perspective, because the happier our volunteers are, the more great science we do, the more impact we have, and the happier we all are. And so I really think we're in a great position to continue a virtuous cycle here. And I want to thank again everyone for their help. I apologize if there were people that should have gotten shout outs that didn't. There's been so many involved that it's hard for me to keep track of. So just thank you so much. And I will call it a day here and look forward to continuing to work with all of you.